จากนี้จะเป็นการบรรยายโดยผู้เชี่ยวชาญจากบริษัท Dassault System นะคะดร.อนันต์คริสนามาตี้นะคะท่านดำรงตำแหน่ง Senior Manager นะคะในส่วนที่ปรึกษาและผู้เชี่ยวชาญซอฟต์แวร์ BIOVIA จากบริษัท Dassault System ค่ะปัจจุบันนะคะมีประสบการณ์นะคะเกี่ยวกับอุตสาหกรรม Life Science มานานกว่า23 ปีเลยนะคะและได้จบการศึกษาระดับปริญญาเอกจาก Biophysics University of Madras นะคะ
So the title is Breaking Boundaries of Drug Discovery with in Silico Approaches. And what you see on the right side is a drug molecule that is bound to a protein and what are its interactions. So we will see what are the different capabilities that is available here. And then I will walk you through some of the great publications that has come out of this, and also some useful case studies and blogs that you will have access to shortly. When it comes to drug development, there are a lot of challenges. First challenge is the increased cost in the R&D. Then second one is how many drugs come to the market. When there is so much scientific artificial innovation, how do we improve the human health? These are the three challenges that is faced. And if we look at these challenges, when we take the data from top 20 pharma companies, they spend 60 billion per year to come out with a drug to the market. And the average cost for one drug into the market is 2.6 billion. 10 years back, it was 140% less, which means that it is increased to 140% as far as bringing the drug to the market is concerned. So what is the answer to this? How do we work on this? We have to collaborate. So how does we collaborate? As academicians, you collaborate with different pharmaceutical industries. You collaborate with different companies like machine learning companies and artificial intelligence companies. So the collaboration is the key to success. And that's what we will be offering to you shortly. When I'm going to take the drug development process, there is a process, there is a strategy which is followed, and above that, there is an artificial intelligence which is applied to this. So what you see on the slide now is the development process from the target validation to the assay to the compound screening till the clinical trials. There is also an addition to that, which is called repurposing of drugs. So you have seen in COVID-19 how drugs were viral drugs were actually repurposed for specifically for COVID-19. So there is a lot of advantage of repurposing of these drugs. And these organizations, pharmaceutical organizations, the AI organizations, academicians go with certain strategies. The strategies are target-based drug discovery, phenotype-based drug discovery, identification of new therapeutics uses. Say, for example, some other viral infections like flu, those drugs were actually used for COVID-19. That's one of the examples. So how do we do this using in silico approaches is what we will be seeing. And there will be an outcome associated with each of these strategies. And that outcome is what we are looking at. When I have a target, I have to validate the target. When I have an assay, I have to develop the assay. When I screen it, I have to do lead identification, lead optimization, and lead validation. So how do I do that? All of these can be done using in silico approaches, which also includes machine learning and artificial intelligence. So AI in drug development or machine learning in drug development or the in silico approaches in drug discovery and development is phenomenal. And today it is the market that we have to go behind. To do that, BIOVIA offers a set of proven protocols and collections. So when you're working with, say, 1 million molecules and compounds and you want to identify that, you go and ask the chemistry collection from BIOVIA. That's very strong in identifying the right compound. You're working with next generation sequencing, the genomics, the mass spectrometry, the proteomics. You ask for the biology collection within BIOVIA. That will help you to do all the proteomic identification, biomarker identifications. Then we have an imaging collection. I will give you some of the examples of imaging selection very soon. You have a machine learning collection. You have a document and search analysis collection. That's not part of the scope of this particular presentation, but we will stick on to the first 4 of them now. So what does drug discovery include? First step in drug discovery, I want to know what is the target. So for COVID-19, it is spike protein, which is the target. So how did I identify that target? Using sequences, using homology modeling, finding out the mutational form of that using protein design, performing simulations. These are the areas where target discovery plays a very, very important role. And the second part is lead identification. That is identification of the drug molecule or lead compound basically. It is based on docking, which is called virtual screening. It is based on a filter using ADME properties or toxicity assessment properties or TopCat properties or database searching. Similarly, lead optimization also exists, and lead optimization is also, lead optimization is also important to optimize a lead compound against a particular target. So these are the inclusions in drug discovery, and this is what in silico approaches helps us to do and arrive at good lead compounds. What does Discovery Studio give you? Discovery Studio gives you a lot of well-validated and acclaimed programs, from homology modeling to sequence analysis to antigen antibody studies to protein engineering. And on the right hand side, you will see all related to small molecules or drugs. QSAR, the ADME, the toxicity prediction. These are the different areas which actually drive the scientific explanation from target to the lead optimization. So the whole process which I showed you in earlier slide is available within BIOVIA's Discovery Studio. Why should you use BIOVIA Discovery Studio? You should use BIOVIA Discovery Studio because they come from very premier institutions and companies. Force field simulation comes from CHARM, that is from Martin Karplus lab, who is the only person who got Nobel Prize in computational chemistry in the year 2013. So you can blindly use CHARM. It's a well-validated program that's available within Discovery Studio. NAMD Molecular Dynamics from University of Illinois, Modeler from Andrew Sally's lab, protein docking from ZDoc from Professor Zhang's laboratories, University of Massachusetts Medical School, protein aggregation from Novartis and Massachusetts Institute of Technology, pharmacophore catalysts from UCSF, and so on. So it's all well validated. For 30 years, we have an association with these premier institutes. First thing, what everybody wants to know and wants to have is a good visualization. How good I see the molecule, how many ensembles of molecule I can see. This is a background image, direct shadow, because we want to have very good publications with very good impact factors. So you need to have good visualization. So Discovery Studio has its own visualization that is available for you to see in different formats. This visualization is not limited to only seeing, but also gives you some useful information. What you see on the right hand side is called Ramachandran plot to study the conformation of secondary structure for stability of protein structures. So that's a very good tool that's available to study the stability of the secondary structures or the stability of the protein. If 88% are stable, the protein is stable. There is a publication associated with that. You can find the secondary structure, you can do a lot of other property calculations. So information rich molecular views are available. You can generate surfaces of these proteins and color it based on different properties. Say, for example, on the left-hand side, I have a surface which is generated based on acceptor and donors of hydrogen bonds. So based on this color and the picture, I can tell what type of compound will go and fit there as a perfect fit. On the right hand side, you see what is called as a mesh-based approach of visualization based on hydrogen bond donor and acceptor, seeing the intensity of the presence. You can see that wherever there is a red color oxygen atom, that is the most electronegative atom which you have, it's blue color and vice versa. So these type of visualizations can be done using surface generation and presentation. That's about the visualization part. It's not only visualization that we are looking at, but we are looking at some number crunching calculations. I have a sequence. I want to search the sequence to build a homology model. You can do homology modeling. You can do mutant modeling. You can do antibody modeling. You can do loop refinement. You can do side chain refinement. So whatever you see written on the left-hand side, that and many more can be done with one module called sequence analysis and protein modeling module. So this is what you see. This is all well validated programs which are available in Discovery Studio. The second one is simulation and X-ray. Whenever a molecule that you have and you want to work on that, you want to simulate the molecule at nanosecond level, at picosecond level, you need to know how the molecule behaves. So it could be a protein, it could be a protein ligand, it could be peptide. That simulation programs are available in a module called simulation and X-rays. The next module is very, very important. People want to do what is called docking. Why should I do docking? I need to find which molecule fits to which target. This is called lead identification for a target. There are three methods that Discovery Studio offers. One is called libdock. If you have 1 million virtual molecules and you want to find good molecules for your target, you use libdock. You have 100 molecules, you want to use a program for docking, you use cDocker. You have 20 molecules and you want to use the best program that's available in the world to do docking, you use flexible protein docking. This is how we do different docking procedures using small molecules against a protein target. If I don't have small molecules to dock, I want to design my molecules. You use what is called as fragment-based design methods. Fragment-based design methods gives you a set of fragments. So Discovery Studio will generate fragments, place it inside the protein, grow it within the protein, and also attach different fragments together and give you the best molecule that fits to the protein target. If that molecule is already patented, you can use replace fragment to replace the non-interacting regions to generate a non-patented molecule which is new and you can patent it. So this is how fragment-based design methods work. The next most important and very strong program with BIOVIA Discovery Studio is called Pharmacophore. It is called as feature-based Pharmacophore. It allows you to search database of pharmacophores. It allows you to perform scaffold hopping. It allows you to do docking. with pharmacophores. It allows you to give what will be my target. I have one natural compound. I don't know what is the protein target. I can use a program called ligand profiler that will give me which is the best target. I can predict the activity. When I take it to the lab, I do experiment. Without the experiment, I can tell what is the activity. I can do fragment design. I can filter a lot of compounds to a small set of compounds. These are the different applications of pharmacophores using Discovery Studio. I want to generate a library of compounds. I want to enumerate, basically it's called enumeration. I want to enumerate a large set of compounds. And that compound, I want to say that it is a kinase-based compound. It is a protease-based compounds. If I want to generate that, I use library design and analysis program. Everything is present within the program. The next most important one is my docked compounds or selected compounds needs to know what is the activity of the compound. Activity is called IC50, EC50 or any other activity. This activity can be predicted using QSAR method, which is called quantitative structure activity relationship method, available in both 2D as well as 3D. One more most important thing is whatever compound that I bring into my lead or try to identify a new compound, it should have no toxicity. So I have to predict the toxicity of these compounds before I take it to the next level. There is a program within Discovery Studio called as TopCat. It is approved and used by what is called the European regulations, EPU. And this comes in a format which is called OECD guideline defined format, which is called Organization of Economic Cooperation and Development. All the models you see in Discovery Studio is present on this particular site, qsrdb.jrc.it/QMRR. So this is a well-validated program for toxicity prediction. We have also included some new and most wonderful things. One is called as free energy multi-site lambda dynamics. If you want to generate a co-generic species of compounds, you use what is called as MSLD technique, which is the advanced level of free energy perturbation methods. We have also developed a program for explicitly having membranes beyond water molecules, and these membranes include a lipid-based membrane. What are these different membranes? So what you see down on the image or the video is different membranes and how the molecule behaves within these membranes, the protein ligand complex or transmembrane proteins. What are the different membranes that we support? We support both homogeneous as well as heterogeneous membranes. And what you see, POPC, POP, DPPC, DMPC, and DLPC are some of the membranes that Discovery Studio gives option to select. And this comes from the best optimization program called CHARM. So that is what we have from the explicit membrane. This is at the small molecule level or protein ligand level. Now today, everybody is talking about vaccine, everybody is talking about antibodies, monoclonal antibodies, and so on. So there is a lot of research which is happening in biologics. Say for example, now for COVID-19, they are working on coming out with an antibody. Already Eli Lilly is come out with an antibody pertaining to that. Like that, many organizations are working on coming out with antibodies, and 100% they have to work with academics to come out with this thing, because collaboration is key to success and innovation. So we will see how we can use the different programs in the biologic space. I have a sequence of an antibody. What all I can do from the sequence of antibody? I can get the motif of the sequence. I can get the conserved amino acid in the antibody structures. I can know what is the antigenic sites. I can annotate the whole sequence using the sequence property prediction which is available. I can generate the structural properties of these proteins. I can also predict the molecular properties of these proteins. I can calculate the hydrophobicity because the core of the protein will have hydrophobic in nature. So more the hydrophobicity, better binding respectively. I can calculate the hydrophobic free energy to determine the stability of that particular protein target or a biologics molecule. Discovery Studio has databases within it. So PDB NR95, NRDB, SwissProt, UniRep, antibody variable. So you don't have to go to internet and download. Everything is present within the Discovery Studio program itself, and you can use them. When you do homology modeling, there is a flow of homology modeling, workflow of homology modeling which is available. And these workflow include some restraints and constraints which is automatically added by the program. That is what you are seeing in this particular slide. Homology modeling with modeler, and the workflow is given on the right hand side. Then you have a set of protein structures and you want to identify the conserved residues and the class specific residues. So with a similarity which varies from greater than 35% to 25%, you see that the conserved residues are the same. The class specific residues are different. So to find the conserved residues in a protein structure, you can use this particular program to analyze the protein conservation pattern. This program is generated by Oliver Fitzgerald's lab in Baylor College of Medicine. Once you build a model, you can do different methods of refinement. The refinement can be loop refinement, can be side chain refinement, can be a small cycle of molecular dynamics minimization. So this is very, very important when you build a model. Now, we have seen the first part of biologics, which includes any biologic, any therapeutic protein. The second part of biologics is antibodies. Antibodies are very important to make sure that we generate the right monoclonal antibody to combat a particular disease. And COVID-19 is something people are working on. People have come out with monoclonal antibodies. We will see what Discovery Studio will offer to you. You can select the right antibody, that is the target selection, predict the activity of this antibody, predict the aggregation, viscosity, and solubility of this antibody. You can calculate the developability index. You can generate the structure of this antibody. You can get the isoelectric point. You can get the dipole moment. You can compare with other antibody sequences. These are the programs that's available to design an antibody from the target perspective. The second one is target optimization and humanization. Most important is how stable is the antibody. When the antibodies start and monoclonal antibodies come in, we have to see at what temperature, what is the stability, at what pH, all this information is required. So a formulation property prediction is extremely important to ascertain this. And you can do protein-protein docking to find the antibody antigen binding capabilities in this particular complex. You can do other property calculations also by predicting disulfide bridges, mutational stability. I will give you some examples as I go on. And then comes the most important thing with my formulation. What are the properties that I am predicting, and is it suitable for this particular antibody or not? So these are the things that I design my antibody therapeutics with. And first part of it is called antibody humanization. And I search antibody humanization is humanizing and identifying the hotspots in an antibody sequence from a set of germline sequences. That's antibody sequence. Then Discovery Studio also helps in what is called the modeling bispecific antibodies. Okay, you can do bispecific antibody modeling using Discovery Studio. That's one of the greatest features that you have with Discovery Studio. Then modeling bispecific monoclonal antibodies. This is a hot topic everywhere in the world today, and people are working on identifying these bispecific monoclonal antibodies and designing them to study their expression, to study the sequence liability, stability, developability, and so on. The next topic is, once I have the antibodies, I have to find when does it aggregate and what is the sequence of aggregation associated with that, and what is the solubility of these aggregated proteins or non-aggregated proteins. So developability index is how these proteins aggregate. If I find the regions of this aggregation, I can do mutations on this region and take the molecule forward. That is why protein aggregation and developability is used. First part of it is, after I find the aggregated proteins and amino acid, I have to do in silico mutagenesis to find the protein stability. Virtually, I can mutate all the residues in the aggregated regions and find what is called the binding and stability by calculating the delta G mutational energy. The lower in the energy, the stable the molecule. So that is what we do using virtual mutagenesis. Then for stability you have done. Now for binding affinity, when you have antigen antibody or a small molecule and macromolecule together, what do we do using protein binding affinity? For that, you can also do that using alanine scanning or amino acid scanning. This is one of the benchmark that we have done with other softwares, and we found that Discovery Studio gives very good results of 78 and 70% accuracy with a least RMS error of 1.1 in the delta G mutational energy calculation. So this is what we do for validation of single mutation binding energy. You can also predict the different formulation properties, which I said you some time back in the first slide, and that will be very helpful for you to understand the monoclonal antibody that you have designed, modeled, humanized, and also identified the in silico mutagenesis for both stability and binding to be the best one by you predicting the formulation properties, and you will see the formulation properties which is shown over here. You can see that we have done it for some monoclonal antibody transmutazole, and all the properties and values related to them agrees with the original one what the organization has come out with. So to summarize, with a protein sequence, you can do post-translation modification, biophysical property calculation, annotate antibody domains, align, compare sequences, and with protein structures, you can generate 3D models, curated antibody structure database, calculate pH-dependent protein properties, predict developability, I showed you, aggregation propensity, I showed you also the in silico mutagenesis for binding and stability, and it can be both on pH dependent and non-pH dependent, which is called molecular switches. So these are the tools for biologics that I showed you a list of them with a description of them sometime above. Now, what is inside these programs? Basically, what do I give inside these programs? Inside these programs, I give you what is called as a workflow, a pipeline. The beauty of this is you can make your own changes. You can make any boxes, change the boxes left and right, up and down, anything. And according to your workflow, you can make your own workflow, which will be very unique and new discovery. That's how we do it. And many organizations do that. This is what it is there. It's totally transparent, totally flexible for you to put your ideas inside this and come out with your own programs. Data science. So it's not only the software in Silico Approaches, but there is a data science related to drug discovery. One of the first drugs to get into the clinical trials comes from a company called Essentia. They use data science to come out with that. And in the next 3 years, 4 years, you will see machine learning, artificial intelligence, imaging to be one of the key concepts or key approaches by which drugs come into this particularly market. Okay, so we will see what it has to offer. So what you see here is a simple imaging technique which allows you to identify images. You have identified a protein, you identify which is the active site of the image. You can do your own machine learning in this particular program. Select those particular regions that you want to identify, make them as positive regions, negative regions, active site regions, non-active site regions, and then run this machine learning program. Then it will predict and give you those specific regions by drawing circles across that. So this program is also available with BIOVIA. Then the next program is model learning and inference using what is called as deep neural network based learnings, where you can basically take the image, identify a particular type of tumor to be benign tumor or malignant tumor. Imagine if I'm going to give you 100 images and you do visual inspection with your eyes and do it, you will take 5 days, 10 days. This program does in matter of few seconds. So if you have 1,000, 10,000, 1 lakh images, it will finish it in less than 1 day. That's the opportunity that you have with imaging. This is one of the publications using imaging for antibody model for excipient interactions for use in computational formulation design. This was done by Massachusetts Institute of Technology, a recently published one in 2020. They have a new biologics monoclonal antibody that predict the aggregation viscosity, model the behavior of the formulation, mutate wherever it is required, and again predict the aggregation and viscosity till they get the correct formulation. So this is a wonderful publication. I recommend you go through this if you're working on biologics or antibody modeling. Another publication from a top pharma in US, North America, and they tested with other different softwares, and they found that the best performing predictor for a data set is Pipeline Pilot, which is BIOVIA Pipeline Pilot. And this using a recursive partitioning model, they were able to find the stable and unstable asparagine and asparagic amino acids in the variable region of monoclonal antibodies, and what is the successor to that. So this degradation sites in antibodies, variable regions is what they predicted. This is one of the wonderful publications from a big pharmaceutical company using BIOVIA. BIOVIA is just not limited to what I showed you on the presentation, but there are a lot of resources that is available within BIOVIA. There is a site for BIOVIA blogs. You can directly type BIOVIA blogs in Google and you will get it. If you type case studies in BIOVIA, you will get all the case studies in BIOVIA where different academics, different organizations have published their case studies. There are 17,462 references which are available inside our site. You can go and fetch them And if you need any specific other one, you can connect with Sigma. The contact details are on the right hand side, and they will help you get more and more publications through either me or any of the other resources we have. I also put up a nice podcast in artificial intelligence for machine learning, which is around 15 minutes, and that's a wonderful one. If you find time, you can also Google. It's available on Amazon, Spotify, and any of the other sites with respect to podcast. You can have a look on that, and that's a wonderful one. You also have a set of YouTube video tutorials for Discovery Studio for drug discovery in Silico Approaches. There are 26 different YouTube videos which is available, each between 40 minutes to 1.5 hours. You can go through those tutorials, and that tutorials will help you to understand how you can discover new molecules, identify new molecule, design new molecule, develop monoclonal antibodies, and so on. So those are the things that is available. And of course, we are always here around the globe, and at your convenient time, you can contact us at the email IDs that I have put up there. So p.chainith@sigmasolutions.co.th and varisara@sigmasolutions.co.th. So both the email IDs are available. You can definitely connect with us in case you need more information on this. So that's the end of my presentation. I would request the moderator to please take it forward for any Q&A.